Welcome back to our channel BioLab, and this is VAMSI. In today's lecture we will be discussing about some amazing things, and the session would be a little fun session. So before getting started, thank you guys for all your support which you are showing to the new website which I have launched, and hope you like that design and template. If you have any suggestions for that, you can comment down in the comment box below. So the website is simple: biolab-vamsi.weebly.com. I'll be pasting the website link in the description box. You can go and check anytime if you want. So, and you can also attend a test. I have already uploaded a test on microbes since we have completed this lecture. And also you can access notes from there. If you want to access notes, 5 rupees payment. In the About Us session in the website, you can see how to pay and everything which I have put. So let's get into today's video without any further ado. Today we will be discussing about plant growth regulators. These plant growth regulators are very important in plants growth, as the word says, they play an important role in plant growth regulation. Like the old name of these was phytohormones. They can be called either way, but we use generally plant growth regulators only. Why can't we call them plant growth promoters, or why can't we call them plant growth inhibitors? Why we are calling regulators? Regulators, while seeing the word, you can understand that they promote the growth of the plant as well as they inhibit the growth of the plant. Both roles are there. So that is why we call them plant growth regulators. In short, we call it as Pgrs. Yesterday, in the last session, I have told you that Pgr is also one of the factor for development. Yes, I have said you that Pgrs are also one of the factors for development, since they help in growth of the plant. So let's discuss what are these Pgrs today. These plant growth regulators or Pgrs can be classified into two types on the basis of their functions, like on the basis of inhibition and promotion of growth. Let me give you Pgrs. I am simply writing. On their promotion and growth inhibition activities, they can be classified into two types. These two types are: the first type is called as plant growth promoters. Plant growth promoters, PGPs, and the second one is called plant growth inhibitors. They are called as PGIs. So these are the two groups: plant growth promoters and plant growth inhibitors. As their name suggests, both promoters promote growth. They are involved in growth promotion activities. They are involved mostly in growth inhibition. These are involved in growth promotion activities, and these are involved in growth inhibition activities. So these are Pgrs. Now let's see that what are these Pgrs. Like what are the examples of these Pgrs which are found in plants? We have totally 5 to 6 Pgrs which we have to discuss. Auxins, auxins, gibberellins or gibberellic acid. Yes, auxins, gibberellins, and you have cytokinins. These all are involved in growth promotion activities. They are involved in growth promotion activities, so they are called as PGPs, plant growth promoters. We have two. They two are ethylene. It is the gaseous promoter. It is a gaseous Pgr. Ethylene and abscisic acid, ABA. These two are plant growth inhibitors. Actually, ethylene has both plant growth promotion and plant growth inhibition activity. It is involved in both. But plant growth inhibition activities are more when compared to the plant growth promotion activities of ethylene. Okay? Plant growth inhibition activities are more when compared to the promotion activities. Okay, these are the Pgrs. Now let's move on to the discovery of Pgrs. Let's see how these Pgrs have been discovered. Everything in science, the most of the things are accidental discoveries. Everything which we see in science, most of them are accidental discoveries, and our scientists gave a name for these accidental discoveries, which are called as serendipity. Serendipity. This means serendipity is a word which is given to the accidental discoveries made by scientists. These Pgr discovery is also an accidental discovery. When Charles Darwin and his son Francis Darwin were working on coleoptiles of canary gas. Let me write these names. Serendipity. Remember the word which is important. Yes. Charles Darwin and his son Francis Darwin. These two are the scientists who are working on the coleoptiles of canary gas. Let me write the spelling. Coleoptile of canary grass. Coleoptiles of canary gas. Now what is this coleoptile? Coleoptile in general, it means the tip of shoot. Tip of the shoot is generally termed as coleoptile. Tip of the shoot is generally termed as coleoptile. When Francis Darwin and Charles Darwin were working on this canary gas coleoptiles, they accidentally discovered something new, that these canary grass were showing phototropism. What is meant by phototropism? Let me erase this. That canary grass was showing phototropism. What is this phototropism? Means it is the growing of plant or bending of plants towards sunlight. See, if this is the sunlight and here you have grass, canary grass. So let me Take some green color. Green, go to green, yes. Here is your canary grass. Let me just give you some moments. Now these canary grass bend towards the sunlight. Bend towards the sunlight. If the sun is here, say if the sun is here, these bend towards this side. These bend towards this side. So this is called as phototropism. This was observed by Charles Darwin and Francis Darwin while doing the experiment on the coleoptiles of these canary grass, and this they found this as a compound which is doing all these functions, which is serving as which is helping for phototropism. They found a compound in the coleoptiles of these canary grass which was helping for phototropism, and they did not name the compound, and they discovered it and they said to the people. But there was one other scientist who is our friend known as F.W. Wundt, who discovered these compounds. He named these compounds. He discovered this compound which was showing phototropism in the coleoptiles of canary grass, and he did a small experiment. See, the scientist was F.W. Wundt. F.W. Wundt worked on oat seedlings. F.W. Wundt worked on oat seedlings and found that the compound was auxin. The compound which helped in this phototropism was auxin. What happened? He conducted a small experiment. When the coleoptile of these grasses, like the Francis Darwin conducted this experiment, when the coleoptiles of these canary grasses were cut, I cut this coleoptile of this canary grass, and there is no coleoptile now, and they were not showing phototropism. If the coleoptile wasn't present, they were not showing phototropism. And now what I did is, I took this coleoptile and I placed it on an agar block. I placed it on an agar block. Here is a coleoptile. This agar block extracted the material from this coleoptile, and it was now acting as the compound auxin, in general auxin. And when I placed it on the top of these canary grass, they were showing phototropism. By this experiment, you can understand that the coleoptiles have had some compound called auxin, which was later discovered by F.W. Wundt, and they showed phototropism. That led to phototropism. So that experiment is in your NCERT. You can read it when you are free. Yes, that is about the discovery of auxins. Now let's discuss the discovery of the second compound, gibberellic acid or gibberellins. Discovery of gibberellins. We have completed auxins. Let's discuss gibberellins. This was also a serendipity when E. Kurosawa in 1926 was working on rice seedlings. He found a disease in these rice seedlings called as Bacane. In general, it is called as foolish seedling disease. Foolish seedling. And this disease was caused by a fungus. It was caused by a fungus, and this is all being observed by Kurosawa. This disease was caused by a fungus called as Gibberella fusiculae. Gibberella fusiculae was a fungus which caused this foolish seedling disease in rice plants. Now, what did Kurosawa did? When he isolated this Gibberella fusiculae and he extracted the compound from this Gibberella fusiculae and sprayed on fresh live seedlings, they were also attacked by this Bacane disease. They were also attacked by this Bacane disease. If he was spraying on some other plants, the plants increased in length. The plants increased in length, like sugar canes, they increased in their length. We will be discussing in the physiological effects of gibberellic acid. And later, this compound which E. Kurosawa identified was named as gibberellic acid from the name of this fungus, Gibberella fusiculae. Gibberellic acid. Gibberellic acid, and until today there were nearly 100 gibberellic acids discovered, and the most famous one was Ga3. These gibberellic acids were named as Ga1, Ga2, Ga3, Ga4. Like that 100, Ga100 were discovered till today, and Ga3 is the most popular and most common thing which is present in plants. Okay, and interestingly, this was the first one which was discovered. Now let's move on to the discovery of some compound which is very important. Let's discover some compound which is very important. Discovery of Kinetin. This Kinetin discovery was very unfortunate. Before talking about the discovery, I have to tell you one thing about callus. This callus, it is a mass of undifferentiated cells. Yes, we have discussed about differentiation. Undifferentiated cells means not mature, meristematic cells. It is a mass of meristematic cells or undifferentiated cells or unmatured cells. And this callus helped and led to the discovery of this kinetin. How? Let's see. When Spoog and his co-workers were working on this callus of the tobacco plant, when Spoog and his co-workers were working on this callus of tobacco, they found something interesting. And when this callus was isolated from the tobacco and led to growth, it was not growing in a nutrient medium. The callus wasn't growing without adding something. This callus wasn't growing. When they added milk, the callus wasn't. When they added coconut milk, it is a nutrient medium. Coconut milk means, in general, it is the coconut water. In science terms, it is called as coconut milk. And when they added this coconut water or milk, it started to grow. The callus started showing growth. And later, Miller and Spoog identified this compound which was useful for the growth of the plant was other than auxins. In the nutrient medium they also used auxins, but it wasn't growing without this compound. Later they identified it as kinetin, and the kinetin later it was classified under cytokinins which showed impact on the cytokinesis of the cell. These showed impact on the cyto. What is meant by cytokinesis? It is a maturation of the cell. Cytokinesis of the cell. They showed impact on cytokinesis, so they were termed as cytokinins. And unfortunately, kinetin was the first cytokinin discovered. About ethylene and abscisic acid discoveries we will be seeing when I talk about the physiological effects of them. And this is it for today's video. In tomorrow's or the next lecture we will be discussing about physiological effects of auxins and Pgrs. Thank you guys. If you like the video, please hit that like button. And also subscribe to our channel BioLab. This is VAMSI signing off from BioLab. Have a good day ahead.